Sure. Good morning, everyone. I think we're going to go ahead and get started. So welcome to the Viruses and Vaccines Seminar. Today we have two exciting talks. First, we'll have Trevor giving an update on avian influenza genomics, followed by a talk from Dr. Pavitra Roychoudhury, who is a research assistant professor in the UW Department of Lab Medicine and Pathology and an associate in vid here in Seattle. And her expertise is in bioinformatics. She completed her PhD at the University of Idaho prior to coming here for her postdoctoral fellowship, and then is now faculty here. And her expertise is really in using viral genomic epidemiology to understand viral transmission. So thanks to both of you for coming to speak today. And before we get started, I'm just going to go ahead and let you know about the next talk, which will be on Tuesday, June 18th. And this will be Carlos Grijalva, who is a professor of pharmacoepidemiology at Vanderbilt University, and he'll be speaking about studies of respiratory viruses in the United States and in South America. And that will be our last in our series of the viruses and vaccines. It has been a great 2 years having the series for everyone, and we'll hopefully be able to maybe do some short seminars in the coming years, but unfortunately, I think organizationally, it's a little bit challenging for us to continue this now as the pandemic wraps up and also the funding wraps up. So thank you all for joining us over the last 2 years, and I'll go ahead and turn it over to Trevor and then to Pavitra.
Great. Thanks so much, Helen. Yeah, and it's been a pleasure being able to participate in these for the last couple of years. So going to try to keep this brief, give lots of time for Pavitra's talk. So I want to give a little update on what's happening with H5N1 genomics. So right now, we still only have 9 states with confirmed infections in cattle, which is surprising given kind of other things we know about, like prevalence in milk and so forth. But I want to focus today on this genomic analysis. This is work that's really led by Louise Moncla, who's now an assistant professor at UPenn. So if we look at the broader context of H5N1 evolution, we can see kind of originally emerging in Southeast Asia, China, mostly being kind of localized to that side of the world until we get to the Americas in 2020. This introduction that then spreads in North and South America. It wasn't here before that in wild birds, but we see that despite it spreading mostly very much mostly bird to bird, we get these spillover infections occasionally. These yellow dots here are things like foxes and skunks and things that are consuming dead birds that have it. They get sick, but we don't really have too much mammal to mammal transmission, except for sea lions in South America and seals in North America, where there have been sustained outbreaks that have had a large impact on the seal populations. We can also see up here that there was an introduction event from birds into cattle in orange in the US that happened fairly recently within the last 6 months or so. And so if we zoom in here, so this is just showing HA. If we look across different genes, we can see a very similar pattern where we go from these blue dots in birds. These are birds that were sampled in the US. And then we have this transition from circulation in birds to orange here, circulation in cattle. We see the same thing across these different segments in flu. So flu is a segmented genome. These 8 segments are kind of like 8 different chromosomes. They can reassort with one another, which makes complications to kind of do a full genome analysis across a large diversity of things. But because we don't see reassortment within the cattle outbreak, we can just concatenate these genomes and look at what's happening in full genomes in the cattle outbreak. And we get a picture like this, where we can see dated molecular clock outbreak here. Orange is cattle. We get some cattle to human, or sorry, cattle to bird spill back, where these are grackle and blackbird and chicken and things that are associated with these farms. As well as in yellow, these are domestic cats who got sick and died drinking milk on these farms. And we can see that there was this introduction into the cattle population sometime between December 2023 and February 2024, so probably January, so spreading for 5-ish months, which we of course didn't notice for months. And then at this point, kind of fairly widespread. If we look at this, oh yeah, one thing to point out here, that this is this red dot is the single human infection. So this is the only documented human spillover so far. This was a dairy worker in Texas. This is kind of weird in how like it's kind of outgrouping to most of the diversity here. So it's not exactly clear what's happening with this this particular long branch, but this is the only human infection so far, or human sequenced confirmed human case that's been sequenced so far. If we look at state level metadata, we can see most of the samples here were collected in Texas, but this still supports an origin of the epidemic in Texas and spread from Texas to other states. Maybe as this fills in, it will change slightly, but that would be what I'd expect. This is congruent with the initial case detection, initial cases reported in Texas as well. And then we can see some specific amino acid changes associated with cattle adaptation. So the main one being this 631L at site in PB2, which we see here, kind of associated with all of the cattle outbreak. Interestingly, that in human-associated cases, you generally get 627K. This seems to be doing a similar-ish thing, but maybe not exactly the same. We haven't really seen adaptation HA at this point, but we've seen some of these other genes with a bit of adaptation, which makes sense in being able to have cow-to-cow transmission. And so we can look at things like, as kind of more data come in, epizootic rate of growth, movement restrictions, adaptive evolution, and so forth. And briefly, I would like to make a plug here that that there was this really cool work done by Peter Hahn, Leah Storita, and Pavitra to sample commercial milk from the Seattle area and look for H5, detected a putative positive in a carton of milk from Colorado. Pavitra did a hybrid capture, got a full genome sequence out of that, and that's now up on GenBank and available here. And you can see fairly remarkably that it's definitely the most recently sequenced thing compared to the kind of data that's been coming out from the USDA. And yeah, so I thought that was really cool. And with that, I will close. And yeah, really looking forward to your talk, Pavitra.
Thank you. Let me share my screen. Great. Thank you so much for having me. It's a pleasure to present here again at the Viruses and Vaccine Seminar Series, and I'm a bit sad to know that it's coming to an end. I've learned a lot from the seminar series. So today I wanted to talk about a couple of really fun projects that were the result of sequencing done at UW Virology combined with really excellent collaboration and hard work from trainees and multiple other people. So I wanted to highlight two projects in particular where the sequencing data provided insight that led us to go in a different direction to what we expected initially. 1 second, I'm trying to just hide my controls here. Okay. So the first one is a SARS-CoV-2 story, and that's the one I'll start with. In a previous Viruses and Vaccines seminar, I described in detail the testing and sequencing that we perform here at UW Virology. So I'm going to keep the background pretty brief here and just say that we crossed over 5.7 million PCR tests performed for SARS-CoV-2 alone at UW Virology. This is testing done on samples that have come from various places, community test sites, local hospitals, and outside clients. And it's really a huge team that does this, led by Greg Pepper, multiple specimen processing teams, multiple scientists, and a lot of automation, instrumentation, and informatics. For the purposes of this talk, what's really of interest is the positive samples that have come out of this testing. So nearly 500,000 positive samples have been found in the testing that's been performed. And of those 500,000 samples, we have, we were pulling aside positives each week for sequencing. So about 20% of those positives were saved and sequenced in order to do surveillance for and detect what variants were circulating. And this is work that was partially funded by the CDC and the Washington State Department of Health. I've described our genomic surveillance workflow previously, but briefly what it looks like is that, as I said, we gather random positive samples each week. And then these have already gone through PCR. And so we select samples that have a CT value less than 31 to improve the chances of recovering a full genome. The selected specimens then undergo RNA extraction and then library preparation and sequencing. We sequence on Illumina instruments, and then we have workflows in the cloud that perform genome assembly and variant identification. We take the assembled genomes and then annotate them with metadata about where they were collected, when they were collected, et cetera, and then deposit those genomes to public sequence repositories and share information with the CDC and the Department of Health. And so in this process, over the course of 4 years, we've sequenced something like we've deposited about 80,000 sequences. We've sequenced about 120,000 samples all total with clinical trials and all this the other workflows besides genomic surveillance. Most of our sequencing involves the use of whole viral genome amplicon panels. These are panels that essentially perform PCR across the length of the whole 30 kb genome of the virus, generate these amplicons that, depending on the size of the amplicons, may or may not need to be fragmented prior to sequencing. So after these fragments are sequenced, then they are bioinformatically assembled to generate the consensus genome. And I've talked in depth about the different amplicon panels, but yeah, we just use a variety of different panels. And these workflows have been automated on liquid handlers in order to increase throughput. And we've talked also about the use of these data in variant surveillance. So all of the data gets aggregated across the different labs across the US, and then is shown in this graphical format in the CDC's variant tracker. And it allows estimates through Nowcast to project which variants are growing the fastest. But besides interesting lineages and interesting mutations, we're also able to look for these edge cases where you have these samples with a really interesting long deletion or insertion that is of interest. So very early in the pandemic, somewhere in like mid-2020, we were trying out various different amplicon panels to see which one we would take forward. And Amin Aditya was a research scientist who was working in Alex Greninger's lab at the time, and he led a lot of that optimization and testing work. And in the process of doing that, he came across some quite interesting samples that had these large deletions in ORF7A and, spanning quite a big chunk. So one was a 392 base pair deletion, and the other was about a 200 base pair deletion. And that 392 base pair deletion was the longest one that was known at that point, that was identified at that point. And so this deletion began in ORF7A and essentially led to the fusion of ORF7A and ORF7D. And he went on to confirm it by performing PCR across ORF7A and 7B and looking at the size of the fragments. But we were not the only ones in 2020 who reported these kinds of mutations. There were reports from Singapore, from Australia, Bangladesh, and Spain, where deletions of various lengths were identified, primarily in ORF7B. The one in Singapore in particular, Singapore team took that one forward because they found that particular deletion was found in about 20% of samples at one point in time. And so they went ahead and looked at clinical data associated with those samples and showed that, first of all, there were transmission chains that were associated with that deletion, showing that it was capable of, being transmitted from person to person. And with the clinical data, they were able to show that it was possibly linked to milder infection. And so given that we had sequenced quite a large number of samples, we were interested in asking, are we seeing these large deletions in our sequences? And is there a way for us to relate that with any clinical metadata that we have to look at the impact on disease severity? So what does a large deletion look like when we look at our reads? So here is what a single sample, when you sequence it using a short read technology like a Illumina method, looks like. So you take your reads and you map them against the genome. And so occasionally you'll see bits of the genome where reads don't map at all. So you see this hole. And so this could be a deletion. So that's why there's no reads spanning this region. But also, the complication that happens for us is because we're using amplicon panels, you can also get the similar kind of effect if there's an amplicon dropout. So what do I mean by that? Amplicon dropout simply means that if there is a mutation that occurs within a primer binding region, and particularly mutations at the ends of the 3' ends of the primers can lead to this more frequently. These mutations can affect the ability of that amplification to occur efficiently. And so this poor amplification is what is seen as an amplicon dropout. So when we take these reads and we assemble a consensus genome, which is basically saying what is the majority base at each site, you come up with a string of ends, but you may not necessarily know, you don't know whether the ends are because of a deletion or is it because of an amplicon dropout that occurred. So to investigate this further, there are two research scientists in the lab at the time, Garrett Perchetti and Noah Baker, decided they wanted to screen this by first getting all the sequences that we had for Washington State as of 2021. So they pulled up, about 10,000 genomes that we had deposited. And then they went through and screened for contiguous strings of ends. And they focused on that ORF7A to ORF8 region. And they picked this number, about 266 base pair, based on kind of the average length of the amplicons we were using in the lab. And so they started screening for these long strings of ends and found 120 sequences that had these contiguous ends. And then we had to go into the freezers and locate the samples associated with those sequences, and found about 89 that had sufficient quality and quantity to do PCR and Sanger sequencing. And of those 89, we found 23 where we were able to confirm that those deletions were real by Sanger sequencing. So those deletions that we identified ranged from 344 to 411 base pairs, which again, the 411 was the longest one identified at that point. So then the next step was, okay, so what is the effect of these mutations? First of all, can we place them on a global or a regional tree to tell whether these mutations are, more prevalent? So maybe there are other sequences that other labs have submitted that also have ends in that region that could potentially have the same deletion. That's one thing. And also, do we have clinical metadata on some of these samples to know whether they're related to a disease severity? And so this is where we approached the Bedford lab, and we were connected to Cassia Wagner, who's a really talented grad student who was working in Trevor's lab at the time, who's now graduated. And we asked her to, work her next strain magic, essentially. So she started to do that, but she also, in the process of doing so, she came back and she highlighted that, she had observed in the sequences while doing this analysis that ORF8 was being knocked out with these large deletions, but there was another mechanism that was also leading to the loss of ORF8, and that was premature stop codons that were occurring within this ORF. And so when she looked at the different lineages, so this was, it was occurring at the time primarily on the alpha and a few of the gamma and delta background sequences. But then fast forward to 2023, we had the recombinant lineages XBB that were also showing this premature stops in ORF8 that was leading to ORF8 being knocked out. And so that was really interesting. And because the ORF8 knockout through these premature stop codons was actually even more prevalent than loss by these large deletions. And so when you look at the tree, she then tried to see whether these mutations were occurring in clusters, going to that transmission question, and found multiple clusters with sizes ranging from very small to quite large, close to 1,000 sequences, where there were where the genomes contained either these large deletions or a premature stop. She also looked at the other genes and asked whether premature stops occur in any of the other open reading frames, and found that ORF8 actually had a disproportionately large number of premature stop mutations, and was also associated with clusters of larger sizes than any of the other genes. And finally, she did a analysis of DNDS, the ratio between synonymous and non-synonymous mutations, and showed that DNDS for ORF8 for these nonsense mutations was actually greater than 1, suggesting that there's potentially a positive selection for ORF8 knockout. And not just that, but clades that had these nonsense mutations in ORF8 had a faster rate of growth compared to the to mutations in other ORFs. And then when we submitted the paper, it went through peer review, and a very astute reviewer suggested that why don't you look at whether mutations in ORF8 also occur intrahost, and that could tell you whether this mutation is being selected for if you see it arise repeatedly within a person as they're infected. And so what we did was we pulled multiple runs from a given time period that were roughly of the same lineage that was circulating at the time. And then she went ahead and looked at mutations, their frequencies, how their intrahost allele frequencies, as well as how frequently you saw those stop mutations occur in the different ORFs. And once again, she showed that these nonsense mutations occur at higher rates in ORF7b and ORF8 compared to the other genes. And finally, the question that we had originally reached out about is is it related to disease severity? So this was done thanks to a collaboration with the Department of Health and the Bedford Lab. So here they were able to connect the sequences to available metadata for a subset of sequences. So with that metadata available, Cassia was able to show that ORF8 knockout was indeed related to lower rates of hospitalization and death compared to samples that did not have the knockout. And also when adjusted for other factors, the odds ratio was something like 0.8 for hospitalization. So in summary, this was a really fun project, and it showed that knockout of ORF8 occurs on different backgrounds and lineages. We showed that positive selection for loss of ORF8 occurs both at the population as well as at the intrahost level, and that ORF8 knockout is associated with less severe disease. This work is currently published, and so please go and check it out. It was really fun to work on this, although it took us like 3 years to wrap it up. Okay, so now switching gears to a different project and a different pathogen, I'm going to talk about enterovirus sequencing that we did for samples from the Seattle flu study. So I'll just start by acknowledging the Seattle flu study investigators and their various groups, because this was a massive undertaking. It was a multi-year, multiple institution project aimed at pandemic preparedness that really, I think, serves as a model for what is possible given the infrastructure, the technology, and the collaborative relationships that can be developed in an area. So you have heard about, various projects from the Seattle flu study in prior viruses and vaccines seminar talks. So I'm not going to provide too much detail other than to say that this was a huge community study, which had a clinical arm that collected samples from patients visiting hospitals and clinics, but also had enrollment of participants across a wide range of sites. This included schools, universities, and daycares, households, tourist attractions, and public transit, workplaces, and congregate living settings, including homeless shelters, adult and family homeless shelters. And it is data from this particular arm of the Seattle flu study that I'm going to present results. So why study respiratory viruses in homeless shelters? So as people who are on this call who live in this area know, Seattle is among the top 5 major cities in the US when it comes to the number of people experiencing homelessness. And these kinds of congregate living settings are disproportionately affected by infectious disease outbreaks because of both individual, factors related to the individuals who live in these, who spend time in these shelters that include, chronic conditions, untreated mental illnesses and substance use disorders, but also factors related to the setting itself. There's minimal space, there's shared spaces for sleeping and hygiene, and there is not an ability to isolate or quarantine or screen regularly, and also due to the transient nature of the population. So the shelter study spanned 2019 to 2021, and it was aimed at recruiting individuals from 23 homeless shelters in King County. A total of 3,281 individuals were recruited during the study period, and this included both residents and staff. In terms of specimens, a total of over 14,000 specimens was collected from participants. And in addition to nasal swabs, there was also a small number of environmental samples that were collected from surfaces and air. The collected specimens then underwent testing for 28 different pathogens using this open array platform. This was work led by Leah Starita's group at BBI. And this panel included multiple, targets for multiple respiratory viruses. The data that came out of this testing was then processed and converted into counts and prevalence estimates for the various pathogens and displayed on a public dashboard run by Trevor Bedford's group. So for this particular project, the initial plan was to look at EVD68, enterovirus D68 in homeless shelters. So EVD68 is one of the members of this very diverse genus enterovirus, which includes multiple viruses like rhinovirus, poliovirus, Coxsackie virus, and many other enterovirus species. It was first isolated in 1962, and then there were sporadic detections over time until 2014, where it was associated with a very large outbreak of severe respiratory disease in the US. And it also coincided at that time with an outbreak of acute flaccid myelitis. It primarily affected kids, but to this day, there's still very little data from adults, particularly in these congregate settings. So the objective of this project was to describe the epidemiology of EVD68 across these adult and family homeless shelters in a major metropolitan area. So this project was led by another really great graduate student, Sarah Cox, who also recently graduated from the Chu Lab, and she, took the lead on this entire analysis. So Sarah took the results from the Seattle flu study testing and identified swabs that were positive for the EVD68 target from OpenArray. So this included 67 swabs, 46 from participants and 21 from environmental samples. And then those were sent over to our lab where we did hybridization capture sequencing. So I just want to give a shout out here to Kevin Kong and Hong Xie from the virology lab who led the sequencing. Kevin was also responsible for the milk sample that you heard about earlier. I'm hearing some noise. I don't know if there's a question, but I can't see anything. All right. So Kevin has become a real expert on capture sequencing. And I'd also like to give a shout out to JD, who is a research scientist, who's a bioinformatics scientist in Alex Granger's lab, and he developed pipelines that allow assembly of genomes from these types of sequencing runs. So we performed capture on these swabs, and out of 67 samples, we got consensus genomes for 56. But what was surprising was that only 7 of those generated consensus genomes were EVD68. So what's up? So most of those EVD68 positive samples didn't generate EVD68 genomes, but instead generated genomes for Coxsackie virus 821. And so that led us to go back to the manual for OpenArray and look at why that might be the case. And so we plotted. We realized there's 2 CT values. There's the enterovirus D68 target CT value, and there's also a target for pan-enterovirus. And so we looked at the CT values, and sure enough, the Coxsackie viruses that were sequenced were double positive. So they were positive for both these targets. I mean, in addition to these enteroviruses, we also picked up some other non-enterovirus species, but those tended to be mostly samples at like really high CT, where the detection of enterovirus D68 is maybe not real, or it could be a co-infection. So this is why it's accidentally on purpose. So we went back and we pivoted to then say, like, well, let's just look at enteroviruses in homeless shelters. And so when I say enteroviruses, this is enteroviruses, but not the rhinoviruses. So we decided to now pull samples that were positive for not just the D68 target, but also the pan-enterovirus target. And at this point, I want to give another shout out to folks from the BBI. So Peter and Luis. Peter in particular has been really great to work with. He was responsible for identifying the specimens, pulling them, coordinating extraction, and getting those samples over to our lab. And Luis helped us sort of understand what was going on with this OpenArray data. So together with these 2 targets, by looking for these 2 targets, these are designed against 102 target strains. So I don't know if I was clear about this, but the OpenArray is actually the OpenArray target for EVD68 was cross-reactive to 16 other species, including Coxsackie virus 821. And that's why we picked that up. Okay, so now we have round 2 of sequencing that was done for samples that were positive for this pan-enterovirus target. And so overall, when we put that data together, we found that of the samples that were sequenced, we found primarily Coxsackie virus 821, EVD68, and some rhinovirus samples. We were also able to identify rhinovirus. We were able to identify these both from participant specimens as well as from the environmental samples. So putting these positives in the context of the overall homeless shelter study, it's a very small number of samples. There were 14,000 samples plus that were collected, and only we're talking about about 55 that were 56 here that were positive for enterovirus, but we can still learn a lot of interesting things. So all of these samples here most of these samples were collected before April 2020. And this is consistent with the observation that, with the stay-at-home order circulation of most other pathogens disappeared for a long time. The other interesting thing from looking at these samples is that most of them, nearly all of them, came from people who were symptomatic. So what were the symptoms that these folks were experiencing? It was primarily runny nose and cough were the top reported symptoms. And of the people who reported symptoms, almost half of them indicated that one or more of their symptoms prevented them from going about their day, their daily activities. The other really cool thing about the homeless shelter is that this kind of rich metadata that was available. So there was information about where in the shelter these specimens were collected from, which I think is really amazing. And so you could take these genotype samples and then overlay them on these floor plans. So you have sort of spatial information at the level of the shelter. So here Sarah's highlighted the different bunks where the resident was, the resident who was using those bunks tested positive and what virus they tested positive for. When I say positive, this is positive and genotyped. So you can see that here, a lot of the Coxsackie virus spread seems to have occurred in this area, in the men's dorm of this particular shelter. And there are similar maps for the other shelters too. And of course, now you have genomes, so you can take the genomes and then put them on a tree. So here, this phylogenetic analysis was conducted by Amanda Casto, who did use NextStrain to take the EVD68 positive samples, and take their sequences. We went down to 5 because we only used unique sequences for one sequence per individual. And she placed them against, contextual sequences taken from GenBank, and found that all of the EVD68 positive samples that were collected from the homeless shelters clustered pretty closely together. And the closest related sequence in GenBank to this cluster was 2 sequences from the US collected in 2020. Unfortunately, there's no information about which state they were collected from, but they were deposited by the CDC. When we look at the Coxsackie virus 812 genome, these genomes, we had 39 unique genomes from 39 participants across different shelters. So the colors here indicate which shelter the sequence came from. And you can see these all look really tightly clustered. That's primarily because there aren't very many Coxsackie virus genomes in GenBank for this 821. In fact, there was only 29 other genomes available at the time when this analysis was done. So the study also made a major contribution in increasing the number of genomes available for this virus in GenBank. But when you zoom in to this cluster, you do see that there is clustering by shelter. So if you look like at the top here, there are multiple identical sequences in the shelter L, and that suggest that they all had a common source or common introduction. But yeah, having more genomes available would help, improve the resolution of this tree. And so this is for the summary of the second study, this is really the first study of the epidemiology of enteroviruses, and specifically among people experiencing homelessness. I think that it shows the power of surveillance of respiratory pathogens, and also environmental surveillance in a community-based shelter setting. I think it also shows that capture-based sequencing is really useful for pathogen surveillance. There's a bit of a trade-off because capture-based sequencing is not as targeted as amplicon sequencing, and at the same time, it's not as unbiased as shotgun sequencing. But given the right probes and the right pathogens that are used to design the panel, I think it can be really a useful tool, as was demonstrated with the milk sample, because the panel just happened to have H5N1 probes in there. So it was, we could just use the panel straight away to sequence this potential positive. And so with that, I just would like to say thank you again and highlight the work of these two really great trainees, and a special thank you to Trevor and Helen, who have been really kind in letting me collaborate with their groups, and also have been very generous with their time. The NGS team over at UW Virology, particularly Kevin and Hong, and the rest of the team here, as well as lots of folks at UW DLMP and virology in particular, and various collaborators and funders. So thank you.
Thanks so much, Pavitra. That was an incredible overview of those two projects, and I really like how you picked the ones that where the sequencing really sort of changed the trajectory of the story and the findings. I'm going to go ahead and open it up for questions, and feel free to just unmute and ask the question. Maybe while we're waiting for others, I'll just go ahead and start. So looking at the shelter maps and the genomic epi of the cases in the shelters, is there a public health intervention that you would be able to use this information to recommend?
Yeah, I think I kind of went through a lot of that pretty quickly. So one thing I didn't mention was that we were also able to recover genomes from environmental samples, and some of them were things like doorknobs and like the women's restroom and things like that. So I think what the data shows is that there's there's respiratory viruses, so like the closeness of the setting really does matter. So I think I'm not sure how to incorporate it in a real-time manner, but I think that there's obviously a lot to learn in terms of like, there's a lot of transmission that happens sort of bed to bed and like bed to, from person to surface, potentially back to person. So I think guidance can be provided, but like I really defer to public health folks on the call to like, how do you implement that real time? I don't know.
Maybe have people in separate rooms instead of in bunk beds all in one big common room.
Yeah, but also like, is it feasible? Like is it feasible? Like they're designed that way for particular, because of the space limitations. So I don't know what's actually feasible in these settings.
Yeah. Looks like Jeff has a question. Go ahead, Jeff.
Oh yeah, hi Pavitra. Great talk, or great talks. I had a question about the ORF8 deletions. Can you elaborate on the function of ORF8 and why you think it had different pathogenicity when it was dropped out? And do we have any thoughts on, or you have any thoughts on that, or any data on that?
Yeah, so ORF8 is thought to, both ORF7 and ORF8 are implicated in sort of interferon responses and immune evasion. And so reduction, like a knockout of ORF8 is therefore thought to, lead to less severe symptoms. But yeah, I think probably, that's what I know so far.
And the driving factor for the deletions is it allows the virus to be more transmittable?
I think the fact that it's associated with these larger clusters seems to suggest that it has a fitness advantage. So it allows it to spread more effectively while causing.
And have you guys been able to measure what that fitness change has been? Is that possible?
No, I mean, just from the rate of growth of those clusters with the deletions, which shows that when in the clusters that have ORF8 knocked out, those clusters seem to grow faster. So sort of indirect measurement, but not But we haven't measured it in the lab, like grow out virus or anything like that.
Great, thank you.
I was wondering if I might ask a question of Trevor about the avian influenza.
Yeah, sure thing.
Trevor, do you think that the reason why we have the genomes from the human and the cattle cases in the US is because we have surveillance here? Why are we not seeing this type of spillover in other regions of the world? Is it because it's not happening there, or is it because we're not looking? I don't think we're
Yeah.
looking here either, which is why I'm a little puzzled why we're seeing it in the United States in a place with such poor surveillance.
No, that's a really good point. So I think there is a real possibility that this 2344B clade, that is the thing that arrived into the Americas, is somehow kind of a bit better at mammalian infections. Like we see more of these foxes and skunks and so forth with it. That's not just limited to the Americas, but it's much more predominant here. It hit the birds here and caused a large kind of epizootic in the birds. So there's kind of maybe more potential in the Americas. But yeah, other places in the Americas, like it could have got into cows, and I guess we might just not know it. And there's also like hints here. I think I can reconstruct this where like I think it was in Poland like last year, there was a number of domestic cats found dead with H5 and they never figured out what was going on. And it could have been some more, more isolated spillover to cattle in that case in some very similar fashion. But there's never detected from cows in that case. So yes, so I think it's potentially that we're just missing it elsewhere, but I think it's hard to say.
Is there milk surveillance in other countries?
Not that I'm aware of, but that would be the cheap and easy way to do things. Clearly, though I don't know how large of an outbreak you'd need, and how much like the U.S. kind of agricultural system has contributed to it spreading so quickly here versus other places things I don't really understand.
Trevor, this is Judy Wasserheit. Thanks for that great presentation. When I think about industrial farming, I think about cows and pigs. And I'm wondering whether you can say anything about, from a genomic perspective, whether we should be looking at other industrialized farmed animals. In the past, flu in pigs has been a threat. In fact, one of the CDC directors got fired over it a number of decades ago. But what are your thoughts about that?
Oh, yeah, this is like I think beyond my expertise in virology for how like if we can actually know from the current makeup of H5, et cetera, all the different segments of whether we can expect circulation in pigs or spill over into pigs. Obviously, we haven't had detection so far. We should be looking. I think the thing that was concerning here was a preprint that was just out from Richard Webby's group that I suspect many people have seen, showing that in cow udders, you have expression of both alpha-2,3 and alpha-2,6 sialic acid, which is like traditionally why we are so worried about pigs, because they express both, and that like gives you a bridge from bird-like preference to mammalian-like preference, or human-like preference. And so it seems like maybe the cows could just do it without have that kind of switch without pigs as well. Yeah, but I don't know functionally what to expect in terms of other animals here.
Thank you.
Any other questions from the group? Okay, well, thanks Pavitra and Trevor for speaking today. It's always great to hear about the updates and viral genomics. And thanks to everyone for coming, and we'll see you again in a month. So
thank you. Thanks, Pavitra.
Thank you all. Fascinating.